Great. All right. Thanks everyone for joining. I'm going to hand over to Michael to give the introduction for our speaker today. So over to you, Michael.
Good day, everyone. Thanks for coming along, Cayelan. We've got Professor Cayelan Carey from all the way from USA. Pretty excited to be here. So if you see her around, make sure you say good day. She's a freshwater scientist, and she's the research is sort of looking at how human behaviour is affecting the lakes and reservoirs, and then in turn sort of how the change in the lakes and reservoirs is then affecting human behaviour. So a bit of a cycle. So a lot of it's looking at ecosystem science combined with sort of data science. So that seems pretty interesting. So we're looking forward to the presentation. So yeah, I'll let you shoot on with it. We'll save the questions to the end just because that's easy, I think, and to pop up. You'll feel free to put your questions on the chat during the presentation, but we'll answer them at the end just because that's working a bit easier with the speaker. All right, thank you.
Awesome. Thanks, Michael and Caitlin, for organizing all this, and for all of you for coming to my seminar today. It's really special to finally be able to arrive into Perth and to visit UWA, and I'm really excited to have the opportunity to meet with many of you while I'm here. So today I'm going to start with a brief overview of the breadth of the research in my lab, and then I'm going to focus in on two ongoing projects. The first is on freshwater biogeochemistry, which involves the analysis of literally millions of data points that my lab has been collecting during the past 9 years. And the second integrates all of this kind of massive empirical historical data with new data science tools to try to predict the future. And finally, at the end, I'll share a little bit about what I've learned along the way, as well as future directions and kind of crazy ideas that I have for moving forward. And you'll notice right here that I have reflections listed twice. And part of this is because I'm on sabbatical right now. And so I have been trying to be really introspective and having a broader picture view of the work that I'm doing and its impact. And just to show you a little bit about where I'm from, you'll normally find me in beautiful Blacksburg, Virginia, which is located in southwestern Virginia, right here within the United States. And we are located in kind of a ridge and valley geography. This is kind of in the central Appalachian mountain chain. We're about a 4-hour drive from Washington, D.C. right here. And within these valleys, there's just absolutely beautiful lakes and reservoirs, which are the focus of today's seminar. So hopefully, now know a little bit more about where I'm coming from and where we're working. So I want to start off by sharing this quote by Thoreau with you, because it really resonates with me for two reasons. So first, lakes are inherently beautiful to behold. So research has shown that just looking at a lake will decrease your stress levels and make you happier. So everyone just look at this picture, take a deep breath, which is just one of the many ecosystem services that lakes provide, in addition to drinking water, fisheries, and many more. And second, lakes are expressive and constantly changing. So because lakes are situated at the lowest position in the landscape, they'll drain their catchments. So they integrate all changes that happen on land due to human activities, and thus truly serve as sentinels of land use and climate change happening on Earth. And so I think truly are acting as Earth's eye. And unfortunately, due to human activities, we're seeing an unprecedented degradation of water quality in lakes and reservoirs around the globe. So as a result of climate and land use change, water quality is becoming increasingly variable, resulting in toxic algal or cyanobacterial scums, as you can see here in this photo, decreasing levels of dissolved oxygen or anoxia, high metals that cause taste, odor, and staining problems, and other impairment that makes lake water undrinkable and toxic for fish. These changes in water quality motivate my work as a limnologist. And in case you're not familiar with this term, limnology is the study of inland waters. And it's inherently an interdisciplinary science, bridging multiple different fields to study freshwater ecosystems. And this is one of the things that I love the most about being a limnologist, is that I get to learn from all these separate disciplines to better understand the mysteries of lakes. And so my lab uses tools and approaches from all these different disciplines to kind of broadly understand how human activities are altering freshwater ecosystems, and in turn, how changing water quality will alter human decision making. And I unfortunately don't have time to talk through all of the work my lab is leading, but I was hoping to kind of share some recent highlights that span each of these different subdisciplines led by grad students and postdocs in my group. And my hope is that within the audience of the School of Agriculture and the Environment, that there's at least one thing that I do that can resonate with you. So first, to start, I'm located in a biology department, and my group has a pretty heavy focus on biology and plankton as indicator taxa of water quality. And so, for example, work that recent PhD grad John Dobek led as part of his dissertation was analyzing continental scale data sets of zooplankton. And he found that reservoir management across the US is transforming plankton food webs. PhD grad Mary Lofton conducted whole ecosystem experiments to study how increased storms due to climate change will affect the likelihood of phytoplankton blooms occurring. And PhD student Heather Wander is leading exciting work analyzing how zooplankton migration patterns within lakes are changing globally due to human activities. For the hydrology component, as part of our work studying reservoirs, which integrate both river and lake environments, we spend a lot of time thinking about how water flows. PhD student Dexter Howard has been leading work studying how high flow events, such as from a hurricane, alter organic matter concentrations. And Whitney Wolmer's dissertation is examining how river discharge is a driver of reservoir continua dynamics from kind of the inflows all the way down to dams. Now, humans live near lakes, and so social science is a really important embedded within the ecosystem wing of my group, because we have to understand how human decisions every day are altering freshwaters. And so to understand this better, PhD grad Nicole Ward is working with decision scientists to develop an app with educational incentives embedded in to improve the likelihood that homeowners will use lake-friendly behaviors. We recently deployed this to over 1,000 homeowners in a lake watershed as part of a massive experiment last year. Postdoc Kate Farrell is working with economists to quantify the financial consequences of poor water quality for property values. And postdoc Tibor is leading an educational program to best study how undergraduates learn ecological modeling and forecasting using real environmental messy sensor data sets. Chris and Xiaomei are two students that are actually from the University of Sydney that have been spending the past 3 summers prior to the pandemic working out of my lab, in which they use some of our data to develop a new hydrodynamic model to study how physical mixing within reservoirs will alter water quality variables. Ryan McClure is a PhD student in my group and used this model in his dissertation to study how turbulence near the sediments will alter methane release to the atmosphere. Finally, for chemistry, I'm really interested in studying and understanding how freshwater ecosystems process different chemical elements. And graduate student Alex Gerling and postdoc Alex Hounshell are studying whole ecosystem cycling of nitrogen and carbon within reservoirs. PhD student Abby Lewis is building on this work with integrated kind of ecosystem and lab experiments to study how much organic carbon that enters into a reservoir will bind with iron, which will affect carbon burial in the sediments at the bottom of lakes, which is a really important carbon sink in the global carbon cycle. And finally, I've worked with 3 geoscience grad students to calculate the contribution of groundwater versus water column processes to what happens in the water column of lakes, especially when it comes to iron and manganese cycling, and as well as building kind of sensors to try to capture these very rapid processes that are occurring on the minute scale. Okay, so that's a lot. And it really only scratches the surface of some of the projects that we're doing in the lab, but I'm hoping that kind of research sampler will provide and kind of the understanding of the breadth of my group. And I want to now transition to thinking about 2 projects in depth, and these 2 projects nearly everyone's involved in. It's some in the periphery, and it's kind of the major focus of our research group right now. The first is using whole ecosystem experiments and models to understand the drivers of historical water quality in a local reservoir in Virginia. And the second integrates all of that historical data with new data science tools to try to understand and forecast what future water quality might look like. And the motivation for this work is that lakes around the globe are exhibiting decreased dissolved oxygen levels. This has been shown in kind of multiple syntheses spanning continents and has been attributed to both climate change and land use change interacting together. And while there's a little bit of a debate as to which of those factors is most important, regardless, we're seeing massive decreases in oxygen globally. And I would argue that anoxia, or low oxygen, is perhaps the most fundamental challenge of our time as a limnologist. So these dead zones in the bottom of lakes cost billions of dollars per year in damage to fisheries, habitat, recreation, and drinking water quality. And while there's a lot known about how oxygen will affect habitat for fish and organisms, as well as single elemental processes, we know far less about how oxygen will affect coupled kind of whole ecosystem biogeochemistry, of how all of these elements are changing in concert together and interacting. And I would argue specifically that we need whole ecosystem work because we have to disentangle the effects of oxygen, which is going down, as well as water temperature, which is going up. And so these concurrent changes require kind of approaches to understanding the effects of oxygen that go beyond lab studies or single elemental studies to kind of whole ecosystem experiments. And so the question that I am addressing in this work is, what are the whole ecosystem consequences of anoxia, or low oxygen, on freshwater biogeochemical cycling? And so to tackle this kind of crazy idea, I've been leading for the past 7 years a project called REDOX, which stands for the Reservoir Ecosystem Dynamic Oxygenation Experiment. And this manipulation is we are oxygenating an entire reservoir at the whole ecosystem scale, and then studying how multiple elements will respond. And so I just want to put the caveat up front that I know that many elemental cycles will respond to changing oxygen, but our focus here is specifically on how it was altering carbon, nitrogen, and phosphorus ecosystem budgets. And so as part of that work, we quantified concentrations, cycling rates, stoichiometry, and major fluxes, which included, for example, sediment burial for carbon, downstream export, and emission to the atmosphere of greenhouse gases, namely carbon dioxide and methane. And this work was conducted in a small eutrophic shallow drinking water reservoir that's located near us in Virginia called Falling Creek Reservoir, or affectionately I'll refer to it as FCR. FCR was built in the late 1800s as a drinking water source for the city of Roanoke, which has about 300,000 people, and it still is used as a drinking water source today and is managed by the Western Virginia Water Authority, a water utility that I work really closely with. At the time in which it was built, the catchment was completely in agriculture, and so it has since reforested since the 1930s, but over that time it has accumulated substantial quantities of organic carbon, nutrients, nitrogen, phosphorus in the sediments. Falling Creek is also a GLEON site, or Global Lake Ecological Observatory Network site, which means that we have deployed automated sensors kind of throughout the reservoir, which are shared with international collaborators. And it's also really important to acknowledge that the land and kind of where the reservoir is situated is on the traditional land of the Tutolo and Monacan communities. Now, unfortunately, cannot take you all to FCR to see this place where I work, so I'm hoping to bring just a snapshot of that to you here. This is just a very quick drone flyover of the reservoir, and my goals of sharing this with you are 3 take-homes. The first is that you can see that it's a kind of completely forested catchment, and those are 2 of my students right there in the boat. The second is that we have deployed sensors kind of throughout the reservoir, monitoring a whole range of different water quality variables. And then finally, it's just an absolutely beautiful place to do science. So I feel very fortunate that I have the opportunity to work there. So we've been manipulating oxygen at the whole ecosystem scale with a hypolimnetic oxygenation system. With this specific type of oxygenation system, we withdraw hypolimnetic or bottom water onshore. We inject oxygen at supersaturated concentrations relative to the water temperature, and then we return that oxygenated water back into the hypolimnion, again that lower layer, at the same temperature and depth from which it was extracted. So when we do this oxygenation, we are not altering water temperature or thermal stratification, and therefore can disentangle kind of changing oxygen from changing temperature. So just to show you what it looks like, this is data here of oxygen in the water column from the first summer we turned on and off the oxygen system. And the x-axis here is time from the beginning of April all the way to the end of September during one summer stratified period. And yes, I realize this is winter for you, but if you can think of it just kind of in the summer stratified time. On the y-axis is depth from the surface of the lake down to the sediments at 9 meters. And the color here refers to dissolved oxygen concentration. So red is anoxic, no oxygen. Blue is high oxygen, 14 milligrams per liter. So at the beginning of kind of our spring period with the oxygenation system off, we had kind of starting out the season after ice came off in the winter, high oxygen throughout the water column. As summer thermal stratification started to set up, where we have a warmer layer on the surface and a bottom colder layer, and we see a separation in the water column, oxygen started to deplete at the sediments down to about 5 or so milligrams per liter on the 14th of May when we turned on the oxygen system for the first time. And when we did this, what you can see is this kind of beautiful increase of oxygen throughout the entire hypolimnion or bottom layer from the sediments all the way up to the middle part of the water column. That stayed high, those oxygen concentrations, until we turned off the oxygen system on the 20th of June. And you can see this kind of immediate decrease of oxygen, especially at the sediments, going down all the way to complete zero or anoxic conditions by the beginning of August. We turned on the oxygen system again. And again, you see that kind of that beautiful linear increase of oxygen concentrations in the bottom part of the reservoir, which stayed high through fall turnover when the bottom and top layers of the reservoir mixed in autumn. So the take home really of what I'm wanting to show this to you here is that we were able to successfully manipulate oxygen at the whole ecosystem scale throughout the entire reservoir by operating this oxygenation system with a switch. And just to show you that we did not affect water temperature or thermal stratification, this is the same time period, except instead of oxygen being shown with the color map here, it's water temperature. Throughout this time, we had a warm surface layer, the epilimnion, and a cold bottom layer, the hypolimnion, which enabled us to isolate the effects of oxygen variability on kind of the carbon, nitrogen, and phosphorus cycling, which was our focus. So over the past 7 years, we have turned on and off this oxygen system on different schedules to really understand under different water temperature conditions how changing oxygen affects the lake biogeochemistry. So we've created a gradient of hypolimnitic oxygen concentrations from summer stratified periods where there's lots of anoxia or kind of no oxygen, as in, for example, the summer of 2018, to also summers like in 2016, where we had really, really super saturated high oxygen concentrations to really kind of compare kind of oxic and anoxic conditions. During this time, we turned on and off the oxygen system for roughly 2 to 5 week periods or even longer over the course of 2013 to 2020 during the thermally stratified period. And during this time, we monitored the reservoir intensively, both with sensors that are deployed and automatically collecting data on the minute scale, as well as manual sampling of total and dissolved fractions of carbon, nitrogen, and phosphorus throughout the water column on approximately 1 meter intervals every week. And this is not work that I did in isolation. This was a massive team effort over 7 years. And so I'm presenting these data to you on behalf of many fabulous grad students, wondergrads, technicians, postdocs, and colleagues who collected the vast majority of the samples that I'll be presenting today and have contributed kind of countless hours, ideas, and feedback that have made this work far better than anything I could have ever done solo. So a priori, we expected that oxic hypolimnia, or when bottom waters have high oxygen, we would have much lower carbon, nitrogen, and phosphorus concentrations than when we have anoxic conditions in the hypolimnia or bottom waters. And this is because when you have anoxic conditions, you have much higher sediment fluxes of carbon, nitrogen, and phosphorus from the sediments into the bottom waters right here. But because there's this kind of suite of interacting biogeochemical processes that are always co-occurring in lakes, it was unclear how the switch to anoxia would affect the overall fate and downstream export of carbon, nitrogen, and phosphorus. And when I say downstream export, I'm specifically referring to how much carbon, nitrogen, and phosphorus that enters into the reservoir from upstream would remain in the system either by staying in the water column or being buried in the sediments or being emitted to the atmosphere, and how much of it is kind of leaves the reservoir downstream. So fundamentally, I'm interested in knowing what is the fate of these elements. Does the reservoir serve as a sink to what comes in upstream, or does it serve as a source of these elements for downstream? So what do we find? So as expected, we observed significantly higher concentrations of total organic carbon, total nitrogen, and total phosphorus in anoxic versus oxic conditions. So the red box plots here show median summer concentrations over the 7 years in anoxic conditions, and the blue box plots show median conditions in oxic conditions during the summer. Now, I don't have time, unfortunately, to show you all of the data on the processes driving these patterns, but the dominant process we found that's responsible for the increased anoxic conditions is release of dissolved organic carbon, release of ammonium or NH4+, a fraction of total nitrogen, and the release of dissolved reactive phosphorus from the sediments into the water column that's really driving these patterns in the total fractions of carbon, nitrogen, and phosphorus. But one of the things that I think is most interesting when you look at this figure is that the responses to anoxia are really different among these three elements. And this results in altered stoichiometry, or the chemical ratios of the system between anoxic and oxic conditions. And what we found was that nitrogen was especially sensitive to anoxia, resulting in much lower total organic carbon to total nitrogen ratios in anoxic versus oxic situations, and much higher TN to TP ratios in anoxic versus oxic conditions. And by comparison, we really didn't see that much of a change, or at least a significant difference, between the total organic carbon to total phosphorus ratios in anoxia versus oxic conditions. And so why this matters, why you should care about stoichiometry, is that the relative proportion of these different elements will control plankton food webs, as well as kind of really fundamental biogeochemical processes such as organic carbon mineralization. So to see such substantial shifts occurring, but not evenly amongst the elements, means that we're going to see really different kind of biogeochemistry in reservoirs as they become more anoxic in the future. So the net effect of much higher concentrations of carbon, nitrogen, phosphorus in the water column is that we're seeing much higher downstream export of all these elements when we have anoxia occurring. So these three figures here show the fate of inflowing carbon, nitrogen, and phosphorus. Any negative value means that the reservoir serves as a sink, a net sink. And any positive value means that the reservoir is net exporting more of the inflowing loads than retaining them. And so what you can see here is that for total organic carbon in oxic conditions, the reservoir serves as a net sink of organic carbon. But in some anoxic summers, we're now starting to see that it can export more organic carbon than it can retain. This is largely because of fluxes of dissolved organic carbon from the sediments into the water column that increase when it becomes anoxic. The reservoir always serves as a source of nitrogen downstream, and it's always a phosphorus sink, but its export of both of these elements, nitrogen and phosphorus, is substantially increased during anoxic conditions. And so in summary, just another way of kind of thinking about this, when the reservoir is oxic, it serves as a net carbon and phosphorus sink, and it exports 95% of the organic carbon that's coming in and 10% of the phosphorus it receives, and it's always a net nitrogen source. However, when it goes anoxic at the bottom, it starts to export far more organic carbon than it retains, resulting in net export downstream. And I think the most striking kind of finding from this work was just how high nitrogen export would increase when we have anoxia occurring, going from about 195% to over 600%, just due primarily to high release of ammonium from the sediments into the water column, with just a smaller increase in phosphorus export downstream. So overall, these data indicate that anoxia will degrade water quality, not just within the reservoir by much higher concentrations of carbon, nitrogen, and phosphorus, but it'll also increase eutrophication downstream, because now we have this much higher exporting load of carbon, nitrogen, and phosphorus that's now in the downstream river that's going into the reservoir that's located below it. So kind of you might think a take home from this work is oxygen is really good. And it is. for water quality. But I do think you can also have too much of a good thing. And so what we found is kind of a side effect of this oxygenation is that when the oxygen is turned on, if you will, at kind of full blast and left on, we essentially are respiring all of that yummy organic carbon that has accumulated over a century in the bottom of the reservoir. And this is resulting in extremely high concentrations of carbon dioxide. So here in this figure is a time series from the end of March to kind of late August, depth again on the y-axis. The color scale here refers to dissolved carbon dioxide, CO2, in parts per million. And for reference, the arrow here, this is at 400 or so ppm, which is atmospheric concentrations. And what we're seeing is that during the summer, where we turned on the oxygen system at extremely high levels and left it on for that whole summer, that we have carbon dioxide concentrations at the sediments that are exceeding 12,000 ppm. So this is really, 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 really high. Again, CO2 concentrations primarily due to organic carbon mineralization of all that oxygen that we're pumping into the bottom of the reservoir. And so the question that we were interested in thinking about looking ahead is, are there approaches that we can use to adaptively manage oxygen levels in the reservoir to both increase water quality to meet the goals of the managers, but also to limit greenhouse gas emissions, specifically to not have to emit so much CO2 from the reservoir? In other words, is it possible for us to forecast conditions in the future when anoxia will occur and preemptively add in just enough oxygen to prevent that anoxia from occurring? And when I say forecast, I'm specifically referring to a prediction of future ecosystem services with quantified uncertainty. And this is kind of an ambitious project that's required a lot of expertise from different team members. And so I'm collaborating with a larger team of social scientists, computer scientists, modelers, and engineers to really think about and develop and study how water quality forecasts can provide vital decision support for managers. And so at a high level, our team is streaming data wirelessly from sensors in the reservoir to the cloud, where we're generating ensemble forecasts that are being used to develop water quality forecasts that can be issued to managers every single day. So similar to a weather forecast, where you might have a prediction of a 95% chance of rain on Thursday, we can make a forecast that says, for example, that there's a 95% chance of a high iron event this Thursday. We do this by deploying a suite of high-frequency water quality sensors in the reservoir that are connected to sensor gateways, which are mini computers that were developed by my colleague, who's a computer scientist, Renato Figueiredo, that stream sensor data to a staging server in a secure way that pushes the data to GitHub every few hours for my team to download. Once the data are in GitHub, we can use them to run open source water quality simulation model called GLM-AD, which stands for the General Lake Model and Aquatic Ecodynamic Modules coupled to them. GLM uses the meteorological data that are coming from the reservoir to simulate lake thermal profiles as well as mixing that's happening in the reservoir. And we've coupled this to modules that are able to simulate plankton food webs and the biogeochemistry to better understand the lake's biology and chemistry. And I want to give a special shout out to Dr. Matt Hipsey right here with his kind of glamour shot photo, who's the developer of this model, and working with him on this project is the reason why I'm at UWA this autumn. So it's been really wonderful to be here with his group and with Matt, just to really be able to think about how best to integrate the GLM-AD model into our forecasting workflows. I feel really grateful for this opportunity. So just to show and summarize one daily time step of the workflow that we use for forecasting, which we call FLARE, which stands for Forecasting Lake and Reservoir Ecosystems. We are streaming model driver data from the field. This is our meteorology sensors that are out at the reservoir, as well as stream sensors on the inflows for the reservoir. These are pushed to GitHub every few hours. We use this driver data to run 1 day simulation with the lake model, the GLM model, which will generate output model temperatures of water temperatures. We can then compare that modeled water temperatures with real measured water temperatures using sensors in the field, which again are pushed to GitHub. We compare that modeled output as well as the observations, and then we use statistical techniques of data assimilation, specifically an ensemble Kalman filter, for adjusting both the model parameters as well as the states. So our forecasts are constantly evolving and being updating over time. This one day of kind of adjusted states are used for initial conditions for the next day's simulation, which we feed into its 16-day weather forecasts, which are coming from the NOAA GEFS, which is a weather product from the US NOAA National Oceanic and Atmospheric Administration's weather product of weather forecasts that have been temporally downscaled for our site. This will then generate 16 days of water temperature and water quality forecasts for our reservoir, which we push to GitHub, as well as use to develop data visualization tools that are emailed to our managers every morning at 7:30 a.m., which is when there's a staff change at the reservoir's water treatment plant. So this is this effort right here is always running in an automated way, generating water quality forecasts every single day for Falling Creek Reservoir. And I want to give a special shout out to Quinn Thomas, located here, who's the modeler that really developed the framework for fitting in observations kind of tied together within a secure cyber infrastructure. So just to show you the example forecast that was generated yesterday, and I realize also that days and months in the US are slightly different from here, but this is for May 3rd, 2022 for Falling Creek Reservoir. And this specific visualization was developed with social scientists that were embedded in the water treatment plant. They actually spent overnight kind of days in the plant trying to understand what kind of visualization was best for the managers and informing their operations. And on the right panel here is the water temperature forecast, which separates out the observed water temperatures for the past week. Here from the surface of the reservoir down to the bottom, what was observed in degrees Celsius. The black line separates the time which the forecast was generated, and then this is the forecasted water temperatures into the future, all the way through kind of mid-May. The mean here of the forecast is shown. This is coming from 440 forecast ensemble members with the confidence bounds around them, which are 95% predictive intervals. Now, the managers care a lot about water temperature because it informs which depth to extract water from the reservoir to the treatment plant. It also gives them an indication of when turnover might happen or when the reservoir might mix. That surface layer and that bottom layer, when they homogenize, usually represents substantial water quality impairment. And so when we're in May right here, we're kind of in solid summer stratification mode. There's not really going to be much mixing that's going to happen. And so the percent chance of a turnover happening is 0. But I want to share with you a forecast from last autumn. So this is from October 13th. Where you can see that the chance of turnover was actually starting to increase right here. Basically, we were leaving kind of that warm summer conditions going into autumn, and the reservoir was going to mix where the surface of the water and the bottom of the water temperatures were getting really close and homogenizing and forecasting that mixing was going to happen very soon in the future. And just to show you why this matters so much to the managers is that there's a lot of accumulated iron in the bottom of the reservoir. And so when it mixes, literally the reservoir will turn red because there's so much dissolved iron that's kind of now all the way kind of through the water column. And they actually usually have to shut down the water treatment plant. And unfortunately, managers will not necessarily know when in advance the turnover will happen because it can happen quite quickly. So here was a water sample taken the day before turnover, and then this was a water sample taken from the surface the day after turnover started during last year. And so the managers simply would not be able to know one day in advance without having forecasts to help them. And again, our forecasting system was specifically focused on being able to forecast turnover substantially or early enough in advance. And so during the past 4 years in which we have been making these forecasts, we have successfully been able to predict the exact day in which turnover would happen about 4 to 13 days prior to that actually occurring. So this is just forecasts of water temperature, but we are now starting to generate daily forecasts of many other water quality variables. And these are providing information for managers that show different oxygenation scenarios. So if, for example, you turn on the oxygen system today, what kind of impact will that have 2 weeks from now? And so these figures show forecasts that were generated on August 15th of last year if the oxygenation system was turned on versus off. And so at the surface of the reservoir, which is right here, 0.1 meters depth, it really doesn't matter if the oxygenation system is turned on or off because of equilibration of oxygen with the atmosphere. So it's kind of thermally stratified conditions. The oxygenation system's at the bottom of the reservoir. It doesn't alter surface oxygen conditions. But at the bottom of the reservoir, so 8 meters, which is about a meter above the sediments, if the oxygenation system is turned on today, which is shown in the blue line, and left on for the next 2 weeks, you can see that there's a substantial increase of oxygen that's occurring in the hypolimnion or bottom waters relative to if the oxygenation system was kept off. And so because of the GLM AED model, we can now simulate a whole range of water quality variables. And so this is oxygen, but we can also start to think about other variables such as methane or CH4. And so this is the same scenario here from August 15th. If the oxygenation system is turned on, you can see that methane concentrations will start to decrease, versus if it stays off and anoxia starts to happen, we'll see an increase in dissolved methane in the bottom waters. Similarly, we can also start to model ammonium, which is, again, really high sediment flux rates of ammonium that can happen in the reservoir, shown from our observational data. And you can see that into the future, we're starting to see an increase of ammonium if the oxygenation system is turned off. And so this is providing both really useful a priori information for not just managing the water quality for the managers, because they care a lot about ammonium for stimulating algal blooms, but also for us and our team about thinking about how to optimize both water quality and greenhouse gas emissions. And in all of these figures here, the solid line is shown as the median of those 440 kind of forecast ensemble members, and the dashed lines here are the 95% predictive intervals. So you can see this work is really just starting to get going, but we're really excited about where it is and its potential. So I'm going to stop for kind of a little bit and pivot and just summarize all of this information that I've provided to you. The main findings we've found from the redox experiment is that anoxia is going to increase carbon, nitrogen, phosphorus concentrations. It's going to alter its stoichiometry in the reservoirs and likely will increase downstream export, thereby kind of not just degrading water quality within the reservoir where anoxia is happening, but also downstream. So inherently it might be creating a feedback by which anoxia could beget more anoxia by kind of transmitting all of that carbon, nitrogen, phosphorus for downstream water bodies. Second, we use the lake kind of modeling infrastructure we developed in redox, specifically that calibrated GLM AED model, to build the flare forecasting system around it, which we're now using to deliver and develop critical kind of information to guide management. So from kind of interviews with managers and our social science colleagues, the goal a priori was to provide forecasts 3 days ahead of time, which is the amount of time that a manager needs to implement changes, such as, for example, order new chemicals or change staffing schedules or put out new filters in the water treatment process. So a forecast of water quality impairment that comes 1 day ahead of time just simply isn't useful because they don't have time to make those changes. So it's kind of not a useful forecast. And so our goal throughout all of this was to be able to provide them forecasts of impairment or changes at least 3 days ahead of time. And so throughout the past 4 years of us being able to get forecasts to them of turnover or mixing events, 4 to 13 days in advance really kind of met our goal for this project. Again, that's focusing again on water temperature, but we're really interested in thinking about adding in now new water quality variables to really help them kind of primarily for managing algal blooms. Kind of finally, my experience working with both FLARE and the redox project indicates that I think forecasts have really great potential for meeting both basic and applied science goals. We are learning a lot about how the ecosystem is working by exploring it in model space. We're also trying to do this in a way that we can provide useful information for the managers that are trying to make these decisions day to day, but are kind of functionally blind for knowing what's going to happen tomorrow. Okay, so from the beginning of our outline, I wanted to kind of dig in deep into these 2 major projects that my lab is running. But I want to kind of now take a step back, go back into introspection mode for really kind of thinking about where we're going. And this has also been really important for thinking about the work that I'm trying to do here on my sabbatical and at UWA. And so a big goal of being here is working with Matt and his team to think about how we can use FLAIR, which is kind of built around the GLM AED model for forecasting new variables in lakes, really thinking about making most generalized software available that can be deployed on a range of different water bodies. And so our team now is received NSF support to deploy FLAIR on 9 lakes across the US that really range in kind of water quality and depth and latitude from all the way kind of from Alaska down to Florida. And so initial work has started to reveal that there might be some constraints to the predictability of different variables, which allow us to kind of answer fundamental kind of ecological questions. So the work that is being led by Abby Lewis has shown that we can only predict about 50% of the variation in chlorophyll about 1 week ahead of time. So this is forecast horizon into the future in days. On the left is R². This is from a synthesis of many different studies trying to forecast chlorophyll or a proxy for algae. And so you might see this and be like, oh gosh, a week out in advance, we can only predict 50% of the variation. But I actually think this is pretty interesting in the fact of like only 1 to 3 days ahead of time, we can do a really good job in predicting chlorophyll. So this kind of work is needed to understand when we can forecast well and when we cannot. And second, kind of preliminary work that's being led by Thomas, in which he has developed forecasts using FLAIR for a range of lakes across the US, has found that inherently some lakes are more forecastable or give, with the same forecasting infrastructure, have much more skilled forecasts than others, which kind of leads to really exciting questions about, are there constraints to predictability that might be related to individual site characteristics, such as lake size, or is it more of a regional weather variable? Are lakes in Florida inherently more predictable than lakes, kind of more north? So this is work that we're really excited about and is driving a lot of the action in my lab right now. The second is my pandemic project with my family has been to build an eddy covariance system in my basement, which we deployed out at Falling Creek Reservoir with my 4-year-old and husband at Falling Creek. And we're now kind of using this to quantify minute resolution greenhouse gases in the reservoir. And I want to show you this picture specifically. You can see this kind of ice cover over the lake. Falling Creek is rapidly losing ice in the winter. And so I'm really interested in thinking about how best to quantify how CO2 and methane dynamics might be leaving the reservoir, might be changing during this ice on versus ice off period. And we're starting to see some really interesting patterns when the reservoir might have ice only on the central part of it, but around the rim where there's water, it could still be emitting substantial methane and CO2 in the winter. And the third is I'm really keen to build on some of the current work that I'm leading as part of Macrosystems EDDI, which stands for Environmental Data-Driven Inquiry and Exploration, which is a teaching program that I run, which has the goal of improving how we teach ecological modeling and forecasting in the classroom. And I've been working with Tig Moore, a postdoc in our group, and we've been trying to develop new tools using interactive Shiny apps for engaging students to really give them hands-on opportunities for introductory students to think about how do you produce a forecast. So taking away kind of all of the coding that you would need for skills, but being able to develop a forecast within an app so that they can think about and kind of really start to understand how variability occurs within real ecosystems. Okay, so most of this talk has been focused on who I am and who my lab group is and what we do and why we do what we do and when. And so I'd like to briefly reflect at the end before we wrap up on how we do our science. And so kind of across all of these different projects that I've provided kind of a sampler for today, I seek to engage experts from different domains. I am truly convinced that having to explain my work to someone from a different discipline enables me to be a better limnologist. It also kind of provides new perspectives, something which I think we desperately need when we're tackling these kind of like wicked environmental challenges. I want to do science that tackles these challenges and helps people. For example, like I want us to be able to predict water quality like we can predict weather. This is kind of a major goal that I've set for our research group for the next 5 years. I realize it is a moonshot and we'll see if we get there, but I think we need to have these kind of broad, ambitious goals if we're trying to solve the world's water quality crisis. I think this is an all hands on deck moment, and I see this really strikingly here in Australia and in Perth, with kind of the changes in water levels that are happening due to rainfall and river flow. Throughout all of our projects, we embrace kind of open source software, tools, and approaches to make our science reproducible. And I'm really kind of always trying to learn new information about workflows for data and code publishing to try to facilitate collaboration, especially across disciplines, but also within research groups. Our lab group spends a lot of time thinking about how best we can help each other be better people as well as scientists. And I think that having this focus on integrating team science and the science of team science into our group is critical moving ahead, especially as trying to integrate and have a full representation of diversity in our group. And finally, I think it's really important to finish on that I want our science to be fun. We are trying to uncover kind of fundamental laws of nature. This is hard. At the end of the day, it is just all about people working and learning together. And I have already learned so much by being here at UWA and from meeting folks and getting a chance to meet and see how teams work here, and feel really grateful for that opportunity. So with that, I just want to thank my lab and reservoir group for collecting all the data that I was able to share with you today. The water utility I work with have been fabulous and really provided some amazing opportunities for working at their sites. NSF, the National Science Foundation, provided funding for the projects. I'm going to give a special thanks to Matt Hipsey, Clarissa Ball, and Audrey Barton, work at the Institute of Advanced Studies at UWA and are supporting my time here with the Glennon Fellowship, and also Fulbright for supporting my trip to Australia. And with that, I would love to answer your questions. Thank you.
Thanks a lot. That was awesome. There's a lot going on in that Sophia there. If anyone's got a question, I reckon just yell it out. It's hard enough. I can't see if you put your hand up on this Teams thing. It's a bit confusing, but yeah. So just see if you can. If you got a question, yell it out. Oh, we'll start with one then while we're getting this all sorted. So obviously, it's a pretty project going on, and part of it with stuff with the lake quality with the managers. You're saying that if you couldn't predict it, then what was happening is that they're just shutting the lakes down, right? So obviously, what's the consequence? What's the consequence of having to shut the lakes down? Is it expensive, or is it just people can't drink for the day, or what's the consequence of having the lake shut down?
Yeah, a great question. Thanks, Michael. So there's a few different things that they do. It depends on kind of what the water quality impairment, kind of how bad it is, if you will. So if there's the picture I showed there where there's like high iron kind of throughout the water column, there's not enough, they can't oxidize all that iron and precipitate it out during the treatment process. So they normally will have to shut down the plant. And unfortunately, when all that iron gets into their system, it'll be shut for multiple days, in which case they have to then change the water treatment allocation. And so then they have to import water from another reservoir into the city. So it's not trivial. There are some tweaks that they can do in the treatment plant. So if they know, for example, that high iron is coming, they can increase oxidizers in the process where maybe they don't have to shut it down for 4 days. It only has to be shut down for a day. Or they can have filters that are coming out so that they can change the filtration system so they don't necessarily have to shut down at all. So it's a combination of both economic as well as staff for them makes that the shutting down the treatment plant is not it's not a good day when it happens.
Yeah, fair enough.
So
I actually can see everyone's hands coming up. So first one up, Caitlin, you've got a question?
I do. Thanks very much, Cayelan. That was a really wonderful talk, and I was writing questions as you were going, and then you'd answer them. So for all the students online, Cayelan's just provided a really fantastic example of how to tell a story about the research, and also very inclusive of all of her students and the people who have helped her along the way. So thank you so much for providing just a really great talk. I was actually quite interested in the forecasting aspects that you've been looking at, and in particular whether or not you're incorporating, and I'm sorry if I missed that in your presentation, whether you're incorporating weather forecasts as well into your forecasting models. Particularly, for example, I'm imagining in areas that are sort of bounded by agricultural systems where you might get a lot of agricultural runoff, because if you were to have, say, an extreme weather event where you had a lot of precipitation and a lot of runoff occurring, how those sort of feedbacks might then affect the oxic and anoxic conditions in the lakes.
Yeah, so in terms of the meteorological forecast, that is, we have done and kind of Quinn Thomas's focus, who's the kind of developed some of the software infrastructure for this, is really on uncertainty analysis. And the single most important or the largest contribution of uncertainty in the water quality forecast comes from our meteorological driver data, as you could probably imagine. And so from, I would say, over 60% of the uncertainty that we have in a prediction is from the meteorology, specifically from wind is a huge one, as well as kind of, I can imagine a reservoir. We also have pretty substantial changes in temperature that happen because it's, the pixel, the statistical downscaling, as you know, is not great. So in terms of this site, we unfortunately it's in a completely forested catchment, so we don't have agricultural runoff coming in, but we are able to get decent, the water driver forecasts, which we have, which are fed by a precipitation model, actually weren't that important overall to the reservoir, despite the fact that it's a pretty short residence time in the water body. It's pretty much all the meteorology. So in terms of thinking about, some of these new sites that we're expanding to, which are, across the Midwest and North Dakota, we've got 2 lakes that we now have flare on. We're finding that they definitely are far more kind of sensitive without having that forested catchment for the inflows. So kind of a good question. I think that's a really, that's what we're trying to get to, right? To scale to these other systems where we can start to test those hypotheses, and as you'd expect, kind of forested versus an agricultural catchment will have a big effect on which driver is most important for forecast certainty. So I don't know if that answered your question, Caitlin, hopefully.
Yeah, it did. Thank you.
Right. Now, I think Amy was next in. If you want to get your question out.
Oh, yes, thank you. Apologies if I missed it in the presentation. It was amazing, by the way, very, very interesting. Loved it. What I was wondering is, would there be, like, in terms of the oxygenation, would there be like a permanent installment of like something that would detect the oxygen levels, and maybe when it hits like a critical level, it's switch on, automates, and then when it hits sort of that, I guess, desired level, it just turns off again, and so you can start instead of having to be continual with it? I'm guessing it's going to, or it's automated already, not sure.
Yeah, so we were talking exactly of where we would like to go. So right now, we do have automated sensors out at the reservoir. We've got oxygen sensors at 3 different depths, and we get those data every 3 hours. So even right now, I can keep an eye on what's going on at home to see what the oxygen levels are. And so that kind of, in terms of where I think you're talking about, which we're excited about, is like event-driven forecasting, and so if, for example, there's an algal bloom threshold that's exceeded on our chlorophyll sensor, it would trigger some management action. Unfortunately, the problem with that is that our oxygenation system is not automated. It requires someone to manually go to the reservoir to turn on certain compressors. There's actually a picture in my talk of one of my students standing on a bucket trying to get one of those, because it's really not, here I can show you very quickly a photo of this. We're not quite at the point where we can do that automated, unfortunately, but I do think, yeah, I don't know if you can see that picture right there. This is Alex standing on a bucket trying to pull the levers that we need to do that. So we can't do that remotely. But I do think that there are other implementations of other systems that are automated, not in the specific reservoir, but other drinking water sources where this could definitely be used. And kind of one of the things I'm excited about is we're starting to work with folks at kind of some of these lakes, large reservoirs out in California, where if we got a forecast, for example, where there's a problem at some depth, it would automatically switch to another extraction depth within the reservoir. So did that answer your question, Amy?
Yeah, absolutely. Thank you.
I think Anas was next. I don't know if I pronounced that right.
I think Amy was next. Oh, no, because it's AG. AG, we have the same initials. Hi, Amy. Anas here. So look, Cayelan, someone says at the start, I don't remember who it was, that a postgraduate student should be taking notice. In fact, that's what I tweeted while you're talking. I tweeted that postgraduate students should be taking notice. Not only that, but everyone should be taking notice of the quality of the presentation, the approach, the inclusivity, all of it. It was just amazing. I'm pleased to see that I think everyone will leave the room, even the people who had no clue what limnology is. And I can see that so many people will be quizzed on that. What is limnology? And they wouldn't be able to answer, but now they know. And I think you made it so accessible. Thank you so much. I really particularly appreciated the fact that you're putting the emphasis on the fun part. Yes. And it has to be about having fun. So, you know, I don't have any particular technical question because everything that you've gone through made sense to me. Especially those like iron thing. I do remember I used to teach a limnology class in Canada, and we used to do like iron, you know, stuff in one of the humic lakes, and it just fantastic stuff. But there is a picture where you are on a Zoom call or a Teams call, and everyone is laughing. I would really like you to tell us what were you laughing about, because it is really important for everyone to understand that the biggest part of science is this thing. This is a book that I keep always close to me. It is about the pleasure that you get from learning together. And with the pandemic and everything, please tell us what you were laughing about. Tell us.
Oh,
you know the one where
yes, yeah,
like the red jumper, everyone is laughing. I want to know what it was.
Yes, I can share this too, for folks to understand the full joy of this right here. Can you see that on us? Yeah. So we have kind of a tradition in my lab of when there's a disagreement, folks have to do a vegetable off. Which is something where you have two people stand, and I mean, these are not serious disagreements, right? It's something like of who's going to go sample today, or so-and-so did something and we need to work this out. But you have two people stand back to back in a hallway, and everyone has to be around, and they have to do their paces, and it depends on how much space you have, but maybe like 3 or 4 or 5 steps, but you need to have enough space, right? So they can be separate from, and their backs to each other, so they kind of pace off. And then somebody within the group who is not part of the vegetable off calls out a vegetable. And then the 2 people cannot look at each other, but they have to act out the vegetable to the best of their ability. And everybody around then decides by vote as to who wins the vegetable off. And I think that was a broccoli that was happening, kind of based by that right there. So that was, and it's really hard in Zoom. Vegetable off. So we've had
a lot of. You gave it a try.
We tried really hard.
I think it's perfect. That's, you know, I knew there was something there, knowing you, but at the same time, I think it's really important for people to just spend more time, you know, having fun and doing things together, and, you know, enjoying and supporting each other. I think it's just fabulous. I mean, I'm so inspired. I'm going to be on a cloud for the next 3 hours. Thank you.
Well, I would love to do a vegetable off with you, or we could do a demonstration here, because I don't think you can truly understand how fabulous they are until we actually, like, everybody in the Zoom room should like do it right now. It's great.
I think we should do it. I mean, honestly, I'm sure someone has a question, but I do remember that there was one time I was in New York with my brother in the family, and we were like, you know, one of those Christmas times where you run out of things to do for fun. And I invented this thing of everyone has to mimic a vegetable. And, you know, it was so funny because nobody knew what vegetables they were doing. So you just do a facial expression, and for you, it's a tomato, but everyone is going to try everything. And you're going to go like, that's a tomato. That's when everyone laughs. But it has nothing to do with the actual mimicking. So I totally relate. We should introduce this vegetable face-off anytime. We should catch up for lunch, by the way.
Yes, that'd be lovely.
Righty, I think That's good. There's a few comments in the chat. A lot of people just saying that they're very happy with the talk, and thanks for doing it. I don't think there's actually any questions as such. So I think in the interest of time, we'll call it a day. And if you've got any questions, you can still stick them in the chat and go back to it after this meeting's ended as such. So if you've got any other questions, just type them in the chat, and we'll see if we can get back to them offline as such.
And send everyone with an assignment to write 1 page about what limnology is.
Oh my goodness.
Right, everyone log off before you hear that. All right. Oh, well, that sounds good. Thanks a lot for that. That's sweet. And thanks everyone for joining.